So previously we studied prokaryotic cells. Now we are studying about eukaryotic cells. Okay. So what is the difference between prokaryotic and eukaryotic cells? Basic difference. So in prokaryotic cells, membrane-bound organelles are absent and nucleus is also absent. But in the case of eukaryotic cells, both are present. Okay. So well-developed organized membrane system is present. Membrane-bound nucleus is present, and other organelles also they are covered by membrane. Clear, Nama? So therefore, the presence of membrane it gives clear compartmentalization of cytoplasm. So what is meant by clear compartmentalization of cytoplasm? It means the different organelles which are present in the cytoplasm, they are separated very perfectly. That is the meaning of that. Okay, ma? So clear separation is present between different organelles in this cytoplasm. So they have complex locomotory and cytoskeletal structures. Similarly, prokaryotic cells they are very simple, but these eukaryotic cells relatively they are more complex. So organelles are also many organelles are present, and similarly the functioning is also very complex. So what is meant by cytoskeletal structures? So a network of interlinking filaments or interlinking proteins that is called as cytoskeleton that we will study in the upcoming slides. Okay. So genetic material is organized into basically chromosomes. Okay, ma? So this is about eukaryotic cells. So then we study about the different organelles which are present in eukaryotic cells. So they contain cell membranes, cell wall, endomembrane system which contains endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Okay. Mitochondria, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus and microbodies. So these are the different organelles which are present in the case of eukaryotic cells. Now the question is, out of them which are present in prokaryotic and which are present in eukaryotic. Clear, ma? See here, mitochondria it is absent in the case of prokaryotic cells. Mitochondria is not present. Clear, nama? So previously it was believed that cytoskeleton was absent in prokaryotic cells. But latest researchers have known that even in prokaryotes also we have cytoskeleton. Okay. So that is out of syllabus just for your information. Clear, Nama? Centrosome, centrioles, they are absent. Clear, no? Nucleus is absent. Microbodies is also absent. In the instead of microbodies, what we have in prokaryotic cells? Inclusion bodies are present. But here what we have? Microbodies are present. Okay, ma? So what are the differences which are present, which are not present? Question can be framed on that. Understood, no? So this total endoplasmic reticulum, Golgi complex, lysosomes, vacuoles, all of them they are absent. Okay. Mitochondria is absent, nucleus is absent, microbodies are absent, centrosome and centrioles are also absent in the case of prokaryotic cells. This is the difference. Okay. So next, first of all, we will study all these parts. All these parts, if we complete, then this completes our topic called cell, the unit of life. Okay, ma? So chemical studies on human RBCs show that cell membrane is composed of lipid bilayer, protein and carbohydrate. Are you able to understand, ma? Cell membrane, the covering which is present on the cell, it is made up of three layers. So a bilayer of lipid is present, protein is also present, carbohydrates also they are present. Okay, ma? So these are the three components which membrane of the cell is made up of. Okay. So here you are seeing a layer, no ma? This total layer, this is called the membrane of the cell, plasma membrane. Are you able to understand, ma? So here you are seeing some small black color structures, no? These structures you are seeing, no ma? These structures. Okay. They are called as lipid bilayers. Okay. And proteins are also present. Similarly, carbohydrates are also present. Okay. So here you are seeing this yellow colored structures. No? They are sugars. They are the carbohydrates. Okay. And you are seeing this red colored structures. No? They are the proteins. And here you are seeing this what you call cylinder type of structure also. No? The cylindrical structure you are seeing, Nama, this is also a protein. Okay, so totally this membrane is made up of three components basically: lipid bilayer, protein, and carbohydrate. Okay, ma. So lipids, phosphoglycerides have outer polar head and inner hydrophobic tails. So the non-polar tail of saturated hydrocarbons is protected from aqueous environment. Are you able to understand, ma? So what is the structure of this? What you call lipid, ma? So see here, you are seeing this pearl-like structures, no here. ఇక్కడ పెరల్ లాంటి స్ట్రక్చర్స్ మీరు కనిపిస్తున్నాయి కదా, సో దే ఆర్ లిపిడ్స్. సో దేర్ షేప్ విల్ బి సమ్వాట్ లైక్ దిస్. సో దిస్ ఈస్ a lipid and two tails will be present. So this is the structure of a lipid. Okay, ma. So this is called the head, called the tail of this lipid. Understand, ma? And what kind of tails they have? They have hydrophobic tails. Hydrophobic tails means what? They are sensitive to water. Water should not be entering into them. Are you able to understand, ma? That is why heads are present on the outer surface of the membrane, and inside this membrane, tails are present. Are you able to understand? See the arrangement of this lipids, ma. So this is the arrangement of the lipids. Okay. So this is the outer structure, and tails are present towards the inner structure because they are hydrophobic tails. Understood, Nama? So head is polar, ma, and tails are non-polar. This is also another important bit which can be asked. Are you able to understand? So head of the lipid is polar, and tails of these lipids are non-polar. Okay, so that is why they are protected from the aqueous environment. Aqueous environment means what? The environment in which water is present. Okay, that's it. So ratio of protein and lipid varies in different cells. Okay, ma. So proteins, the amount of proteins and lipids, they are not same in every cell. For example, if you take human RBC, membrane has 52% protein and 40% of lipid. Understood, Nama? This varies from animal to animal, from cell to cell. Okay. So based on the ease of extraction, how easily we are able to extract the proteins, again proteins are divided into two types. One is integral protein, second one is peripheral proteins. Are you able to understand, ma? So integral protein means what? These proteins are very tough to extract from the cell. That is why they are called as integral proteins. Whereas these peripheral proteins, they just lie on the outer surface of the cell and they can be extracted easily. Are you able to understand? So Seema, here the structure which you are seeing, this is totally peripheral protein. This is just present on the outer surface or inner surface of this membrane. They are very easy to extract. But here, these proteins, these channel proteins and carrier proteins, they are integral proteins. Integral proteins means what? They are present, they are integrated along with the membrane of the cell, and they are very tough to extract. Clear, no? So based on that, we are dividing the proteins into two types: integral as well as peripheral. Okay, ma? Next. So, fluid mosaic model of cell membrane. What is meant by fluid mosaic model of cell membrane, ma? It means that the proteins which are present in this cell membrane, they can have some lateral movement. Lateral movement means what? Lateral movement means sideways movement. So these proteins, they can move like this. Are you able to understand, ma? So this type of protein movement, that is called as a fluid mosaic model of cell membrane. Are you able to understand, ma? It was proposed by whom? It was proposed by Singer and Nicholson. So according to this, the quasi-fluid nature. What is meant by quasi-fluid nature, ma? It means it is not 100% solid, it is not 100% liquid. Such type of materials, we can call them as quasi-fluid nature. Of lipids enables lateral movement of proteins within the overall bilayer. Okay, ma? It means in this bilayer of proteins, lipids, proteins can move laterally. Lateral movement means what? Sideways movement. Okay, ma? So this observation was done by Singer and Nicholson, and that is why this model we call it as fluid mosaic model. So this ability to move within a membrane is measured as its fluidity. So if the proteins are able to move within the lipids easily, then we say its fluidity is greater. If it is not able to move freely, then we say its fluidity is lesser. That's it. Okay. Then functions of the cell membranes. What these cell membranes can do. Okay, ma? So transport of the molecules inside and outside of these membranes. Okay, ma? Membrane is selectively permeable to some molecules present on either side of it. What is meant by selective transportation, ma? Selective transportation means what? So this membrane, it can select what kind of molecules it requires to send out of the cell and into the cell. Every type of molecule, every type of atom, it will not accept. Only those cells which are required by the cell are not required by the cell. Only those are transported. Are you able to understand, ma? That is why this type of transportation, what we call selective transportation. Okay. So this is one of the function of cell membrane. So due to fluid nature, plasma membrane can help in cell growth, okay, ma? Formation of intercellular junctions, secretion, uh, endocytosis, clear, ma? And cell division, etc. So these are some of the very important functions of this cell membrane. Are you able to understand? So it has a fluid nature, no? So it means it is flexible. So because of this flexible nature, it can do many of the functions. Okay, for cell growth also responsible. So between cells, junctions will be formed, so that information is passed from one cytoplasm of one cell to cytoplasm of the next cell. So junctions are also very important. So these junctions are also formed with the help of membranes. Secretion is also present. Endocytosis. What is meant by endocytosis? Transportation of molecules is nothing but endocytosis. And cell division is also possible with the help of cell membranes. So these are the functions which you should learn very carefully. Okay. Next, types of transport: active transport as well as passive transport. What is meant by passive transport? Passive transport means if materials or molecules are traveling from high concentration to low concentration, it is called as passive transport. Are you able to understand, ma? And this does not require any energy. Energy is not required for this one. Are you able to understand? For example, we study in a heat topic. Now, heat naturally flows from hotter body to colder body. This type of transport we call it as passive transport. Here you need not spend energy. But active transport means what? Molecules should travel from lower concentration to higher concentration. Then that type of transport is called as active transportation. And in this condition, ATP should be spent. What is meant by ATP? It is called as the currency of the cell, no? Amount of the cell, the money which the cell has, energy currency. Okay, ma? So this ATP must be spent. Then only molecules can be spent from. They can be sent from lower concentration to higher concentration. Are you able to understand? This is called active transport and passive transport. Okay. And now see here he has given the complete description of passive transport. So movement of molecules across the membrane along the concentration gradient. What is meant by along concentration gradient? Higher concentration to lower concentration without the expenditure of energy. Energy is not spent here. Okay, ma? So therefore it is of two types: simple diffusion and osmosis. Okay, ma? What is meant by simple diffusion? Neutral solutes. If they are moving, then it is called as simple diffusion. Neutral solutes. Are you able to understand, ma? And if water is moving by diffusion, it is called as osmosis. So that is the only difference between simple diffusion and osmosis. If solutes, if they are moving, then it is called simple diffusion. Water, if it is moving, it is called as osmosis. Are you able to understand, ma? So here we have a figure. We will come to this figure later. Okay. Now see, ma. Next, already we have studied, no? This one. So polar molecules cannot pass through non-polar lipid bilayer. Are you able to understand, ma? This bit also is very important. Polar molecules, molecules which have some what you call positive sign or negative sign, they are called polar molecules. Ma. These molecules, they cannot pass through the lipid bilayer. Okay, ma? So only head of the lipid has some charge. No? Tails of this lipid does not have any charge. That is why they are called as non-polar tails. Okay. So through these non-polar tails, polar molecules cannot travel. So what they do is they construct a bridge of proteins. ఒక ప్రోటీన్స్ యొక్క బ్రిడ్జ్ అక్కడ అవి కన్స్ట్రక్ట్ చేసుకుంటాయి. క్లియర్ మా? So they require membrane carrier protein for their transportation. So this type of transportation, what we call it as? We call it as facilitated diffusion. Are you able to understand, ma? So see here what is happening. This one is simple diffusion. Okay, ma? So they are non-polar molecules, and non-polar molecules can easily pass through the membrane. Are you able to understand? But here the molecules which you are seeing here, they are polar molecules. And these polar molecules, they require a bridge of protein. Okay, ma? And through this bridge only, they can enter into the other side. This type of transportation, we can call it as facilitated diffusion. Understood, Nama? And in active transport, what happens in active transport? ATP must be spent, energy must be spent. Then only molecules can travel from lower concentration to higher concentration. Okay? So it is natural that molecules travel from higher to lower, Nama. But if you want to send molecules from lower to higher, then definitely you need to spend energy. Okay, ma? And only by this process of active transport only, cell can discharge its waste. In every cell, some waste material will be produced, no, ma? So that waste material will be transported out of the cell only with the help of active transport. Clear? That's it. Okay, ma? So then we are studying about active transport. So movement of molecules across the membrane against the concentration gradient, that is from lower to higher concentration with the expenditure of energy, ATP is utilized. Example is Na plus or K plus pumping system. So Na means sodium, K means potassium. If sodium and potassium molecules, they are also micronutrients. No? So these micronutrients, if they should be pumped, definitely we should require the process of active transport. Clear? Next, cell wall. So it is a non-living rigid structure found outer to the plasma membrane in the case of fungi and as well as plants. So where this cell wall is present, they are present in fungus also, they are present in plants also. But in animals, this cell wall is absent. Where this cell wall is present, just surrounding the plasma membrane. Plasma membrane will be there. Plasma membrane is surrounded by cell wall. Okay. And what is the main function of this cell wall? Main function of this cell wall is protection of the cell itself. Okay, ma? It doesn't allow unwanted molecules or unwanted things to enter into the cell. That is the function of cell wall. So cell wall of algae is made up of cellulose, galactans, mannans, and minerals like CaCO3. Question can be asked on this. Okay, ma? Cell wall of algae is made up of these things: cellulose, galactans, mannans, minerals like calcium carbonate. Clear, nama? In other plants, it consists of cellulose, hemicellulose, pectins, and proteins. Okay, ma? So these differences of what cell wall is differentiated in terms of algae as well as plants, this difference you should remember. Okay. Question is that where mannans are present. So cell wall of algae, cell wall of plants, cell wall of human beings, cell wall of some other things. Then the answer is algae. So like this, you have to remember these things. Clear, Nama? So what are the different materials present? Carefully you should by heart. Next. So cell wall of young plant cell is capable of growth. Okay, ma? If you have a plant which is present at its initial stages in the bud form or in the preliminary stages of plant, then it has the ability to grow. Once this cell matures, then the cell wall cannot grow. Once the cell wall cannot grow, then that type of wall we can call it as secondary wall. Are you able to understand, ma? And it is formed on the inner side towards the membrane. Are you able to understand, ma? So secondary wall, is it present? It is present on which side? It is present on inner side of the membrane. Are you able to understand, ma? That's it. So what is meant by primary wall? The wall which has the ability to grow is called primary wall. The wall which doesn't have the ability to grow, it is called as secondary wall. That's it. Clear? So then we have a layer which is called as middle lamella, which is present between two cells. రెండు సెల్స్ యొక్క వాల్స్ మధ్యలో. సో దెర్ విల్ బి అనదర్ లేయర్ విచ్ ఇస్ కాల్డ్ as మిడిల్ లామెల్లా. సో మిడిల్ లామెల్లా is a layer mainly of calcium pectate. Okay, middle lamella is made up of which material? Calcium pectate. It glues different neighboring cells together. Okay, ma? So this middle lamella is a sticky substance secreted so that it can hold all the cells together. Understood, no? The cell wall and middle lamellae may be transversed by plasmodesmata, which connects the cytoplasm of neighboring cells. So what is the meaning of this one? So this is one cell, this is another cell. Are you able to understand? Ma? In between these two cells, there should be some connection, no? Are you able to understand? Ma? There must be some connection. Okay, this is the connection. So that connection itself, what we call that, we call it as plasmodesmata. Are you able to see this one, ma? So this connection between two cells, that is called as plasmodesmata. And what this plasmodesmata will do? This will connect the cytoplasm of neighboring cells. So to pass information from one cell to another cell, there should be some passage, no? So this plasmodesmata serves as the passage. Okay. So see in the figure also you can see here. So these are two cells, ma. So this is the plasma membrane. This is the cell wall. Okay, ma. And these connections which you are seeing here, that is called as plasmodesmata. Clear, no? Next. So functions. What are the functions of cell wall, ma? It gives shape to the cell. It protects the cell from mechanical damage and infection. Cell to cell interaction. It means passing of information. Acts as barrier to undesirable molecules. So basically, protection and cell interaction. This is the function of the cell wall. Understood now? Next, endomembrane system. So endomembrane system consists of what? It consists of endoplasmic reticulum, Golgi complex, lysosomes as well as vacuoles. So all of them together, it is called as endomembrane system. Okay. So endoplasmic reticulum, what it does? Endoplasmic reticulum is basically divided into two types, no? Rough endoplasmic reticulum as well as smooth endoplasmic reticulum. So on the rough endoplasmic reticulum, we have ribosomes. Where ribosomes are present? Ribosomes are present on rough endoplasmic reticulum. And these ribosomes are responsible for producing proteins. So therefore, what is the basic function of endoplasmic reticulum? The production of proteins or the synthesis of proteins. Understood, no? Then we have a Golgi complex. What this Golgi complex will do? These proteins which are produced by the endoplasmic reticulum, that is received by the Golgi complex. So these proteins, they are further modified and they are transported to the required locations. That is the function of Golgi complex. Or recently, this Golgi complex was named as Golgi Apparatus also. Are you able to understand, ma? So what is the functions, ma? Endoplasmic reticulum, it produces proteins, and that proteins is received by this Golgi complex. It further modifies this proteins and it is transported to the required regions. That is the function of Golgi complex. Questions can be asked. Clear, no? So lysosomes. Lysosomes, they are responsible for synthesis or the digestion of proteins and different carbohydrates. Are you able to understand, ma? So what is the function of lysosome? Digestion is the function of a lysosome. Okay. Next, vacuoles. So vacuoles are responsible for storage, ingestion, digestion, excretion, and expulsion of excess water. So this is the function of a vacuole. Okay, ma? And the size of the vacuole also, it varies from one type of cell to another type of cell. What is meant by vacuole we will study in detail. Are you able to understand? So in the figure you can see. So this type of structure we can call it as a vacuole. Okay, ma? And how much area they will cover, that also varies from different types of cells. So what is the function of vacuoles, ma? Storage, ingestion, digestion, excretion, and to remove the excess water which is present in the cell. Okay. Lysosomes are responsible for digestion. Okay. So here, this is small yellow color structure you are seeing, no? This is called as a lysosome. Clear, no? That's it. Okay, ma? So, endoplasmic reticulum. So these are a network of tiny tubular structures scattered in the cytoplasm. So we know endoplasmic reticulum is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. So this rough endoplasmic reticulum, it is very close to nucleus. Okay, ma? Very closer to the nucleus. And smooth endoplasmic reticulum is a bit far away from the nucleus. Clear? So tiny tubular structures scattered in the cytoplasm. So endoplasmic reticulum divides the intracellular space into two compartments. So luminal and extraluminal. What is meant by luminal? So that region in which nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, all that region, that is called as a luminal region or inside ER region. Okay, ma? And total remaining region is called as cytoplasmic region or extraluminal region. Are you able to understand, ma? So see the structure of this arrangement of this rough endoplasmic reticulum, ma? So this is the structure. This is how this rough endoplasmic reticulum is arranged. And here the small dots which you are seeing, these small dots, they are nothing but ribosomes. Are you able to understand? So therefore, what is the main point in this one? Here we have to study endoplasmic reticulum divides the cell of the space into two regions, luminal region and extraluminal region. That's it. Okay, ma? So rough endoplasmic reticulum. So they bear ribosomes on their surface. Ribosomes are present. Rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis as well as secretion. Okay, ma? So these are the two functions of rough endoplasmic reticulum. Okay, proteins are also synthesized. Secretions also will be done. ఓకే సో ఇటువంటి వాటిపైన మిమ్మల్ని క్వశ్చన్స్ అడుగుతాడు అర్థమవుతుంది కదా ఎవరు అయితే కాన్సన్ట్రేట్ చేయని పాయింట్స్ ఉంటాయో వాటిపైన క్వశ్చన్స్ వాళ్ళు ఫ్రేమ్ చేస్తారు సో వాట్ ఆర్ ద ఫంక్షన్స్ ఆఫ్ రఫ్ ఎండోప్లాస్మిక్ రెటికులం నాలుగు ఆప్షన్స్ ఇస్తాడు ప్రోటీన్ సింథసిస్ ఒకటి సెక్రేషన్ ఒకటి ఎక్స్పల్షన్ ఆఫ్ ఎక్సెస్ వాటర్ ఒకటి డైజెషన్ ఒకటి అప్పుడు ఆప్షన్స్ ఏమవుతాయి ప్రోటీన్ సింథసిస్ సెక్రేషన్ ఆప్షన్స్ అవుతాయి అలా సో దే ఆర్ ఎక్స్టెన్సివ్ అండ్ కంటిన్యూస్ విత్ outer membrane of the nucleus. Continuous means what? They are attached to the outer membrane of the nucleus. Are you able to understand, ma? Smooth endoplasmic reticulum. Ribosomes are absent here. Okay, ma? It is the major site of synthesis of lipids. Lipids are produced where, ma? Lipids are produced in smooth endoplasmic reticulum. Are you able to understand? Proteins are produced in rough endoplasmic reticulum. So in animal cells, lipids like steroidal hormones are synthesized in smooth endoplasmic reticulum. In the case of animal cells, which kind of lipids are produced in SCR? Steroidal hormones. They are also produced in smooth endoplasmic reticulum. Are you able to understand? That's it. So in the figure you can see, ma. So this total structure you can call it as smooth endoplasmic reticulum, and this total structure you can call it as rough endoplasmic reticulum. And rough endoplasmic reticulum, it is in contact with the nucleus itself. Okay. Next, Golgi apparatus. So densely stained reticular structures near to the nucleus. Okay, but densely stained reticular structures. First observed by whom? Camilio Golgi. That is why its name is given as Golgi apparatus. So they consist of flat disc-shaped sacs. And so what is meant by disc-shaped sacs, ma? So here, uh, these sacs which you are seeing, red color. You are seeing these sacs, no? So they are called as cisternae. Are you able to understand? Okay, ma? So they are stacked parallel to each other. So these cisternae, they are placed side by side. But there are some interconnections also in between. So in between all these cisternae, you can see some interconnections also. So how many cisternae are there? Chala unnai chudandi ikkada 1, 2, 3, 4, 5. So all of them they are called as cisternae with some interconnections between them. So cisternae are concentrically arranged with convex cis face and concave trans face. So cis and trans faces are totally different, but they are interconnected. Okay, ma? So this region is called as forming region. Are you able to understand? And this side we can call it as maturing side. Okay. Forming side means what? So on this side from here, you can see the formation of this system. And once they start growing, they start maturing on this. Are you able to understand? And this phase is also called as cis phase, and this side is also called as trans phase. So are you able to understand? So therefore, Golgi apparatus have two phases. One is cis phase, which is also called as the forming phase or the convex phase. Second one is called the trans phase, which is also called as maturing phase, or you can call it as the concave phase. So these names also you have to by heart now. Maybe questions can refer to this. Okay. So functions of Golgi apparatus. What are the functions of this Golgi complex? Let us see. So secretes materials into the intracellular targets or outside the cell. So this is first function of what you call Golgi apparatus. Okay, ma? Secretion is also a part of Golgi apparatus. Okay, first function. Materials to be packed as vesicles. It means this is the manufacturing center of vehicles. Understood? So already we studied that for transportation of materials we required vesicles. So where these vesicles are manufactured? Vesicles are manufactured in the Golgi apparatus. Clear, ma? So here you are seeing this one structure, this structure. So this is an immature vesicle. Still it is not completely formed. Are you able to understand? Once it matures, it becomes a vesicle ready for transportation. Are you able to understand? So materials to be packaged or vesicles as vesicles from the endoplasmic reticulum fuse with the cis phase and move towards the trans phase. Okay, ma'am? So whatever materials are produced in the endoplasmic reticulum, what are the materials produced in endoplasmic reticulum? There are many secretions also, there are many proteins also. So all these they travel uh, in this Golgi apparatus. From where they start? They start from the cis phase and they reach the trans phase. Okay. So this is why Golgi apparatus remains in close association with the endoplasmic reticulum. That is the reason why it is attached to the endoplasmic reticulum. So proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its transphase. So already we discussed, so the proteins which are produced in endoplasmic reticulum, they are again modified by whom? Modified by the cisternae which are present in Golgi apparatus, then they are released from the transphase. Okay. So, and what is the one more function of Golgi apparatus now? So, formation of glycoproteins and glycolipids. This is also one of the function of Golgi apparatus. Are you able to understand? Clear, Nama? So, totally how many functions he gave? Three functions are there. So, secretion of materials required. Are you able to understand? Transportation of proteins and modification of proteins as well as formation of glycoproteins as well as glycolipids. And one more function is formation of vesicles also. The vesicles which are required for transportation, they are also manufactured in Golgi apparatus. So these are the different functions. Okay, lysosomes. So what is SIMA here? The structure of lysosome is seen here. This is the small lysosome floating in the cytoplasm. So membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. Okay, no? Lysosomal vesicles contain almost all types of hydrolytic enzymes like hydrolases, lipases, proteases, and carbohydrases. They are active at the acidic pH. They digest carbohydrates, proteins, lipids, and nucleic acids. So already I said these lysosomes, they are responsible for digestion. Are you able to understand what they digest? They digest carbohydrates, proteins, lipids, and nucleic acids. Okay, and they are also a type of vesicles. These lysosomes also, they are a type of vesicles only. So they will be moving in and out of this cell. Sorry, inside the cell only, they will be moving continuously, and they will be responsible for digestion of these proteins, carbohydrates, lipids, and nucleic acids. So they contain some enzymes also. What kind of enzymes they have? They have hydrolytic enzymes. Okay. So examples are lipases, proteases, and carbohydrates. So questions can be asked on this particular bit also. Okay. Understood, Nama? That's it. Next, vacuoles. So this is a very important bit where questions were asked many times previous. Vacuoles. So previously we studied the functions of vacuoles. They are responsible for separation. They are responsible for ingestion. They are responsible for digestion. They are responsible for removing what you call the materials which are not required by the cell, they are responsible for removing excess water also from the cell. So that is the function of a vacuole. Okay. So they are membrane bound. So every organelle present in what we call this eukaryotic cell, definitely it is membrane bound. Okay. So membrane bound space found in the cytoplasm. So it contains water, sap, excretory product and other materials not useful for the cell. So vacuole is bound by single membrane called tonoplast. So what is the membrane of the vacuole called ma? It is called as tonoplast. So in plant cells, the vacuole can occupy up to 90% of the volume of the cell. Okay, ma? So there are many organelles present inside the cell. But in the case of plant cell, only vacuole can occupy up to how much part? 90% of the part. The small membrane is present in which water is present, sap is present, and the excretory product produced in the cell is also present. Are you able to understand now? So all of them, they are present inside this vacuole. Clear? That's it. And this membrane is also a permeable membrane. The tonoplast which you are seeing here, this is the vacuole. This total structure which you are seeing, this is called as the vacuole. Clear? So that membrane which you are seeing, transparent membrane, that is called as tonoplast. And tonoplast is a semi-permeable membrane. It can also transport materials outside and inside of this vacuole. Clear? That's it. So vacuoles, same vacuoles we are studying. In plants, tonoplast facilitates the transport of ions and other materials against the concentration gradient into the vacuole. Are you able to understand? So this tonoplast, what it will do, it will transport materials against the concentration gradient. What is meant by against the concentration gradient? It means materials are traveled from lower concentration to higher concentration. That is called as against the gradient. Okay. Hence their concentration is higher in vacuole than in cytoplasm. Are you able to understand now? So in amoeba, the contractile vacuole helps for excretion. In amoeba, what kind of vacuole we have? Contractile vacuole. It means this vacuole can contract. By this process, the waste material is removed from the cell. Understood, Amma? In many cells, example protists, food vacuoles are formed by engulfing the food materials. In some type of cells, example is protistoma, food vacuoles are formed. Now, what is the type of vacuole we are studying? This vacuole contains water, sap, excretory material also. But some vacuoles are present which are called as food vacuole. They what you call, they engulf. Engulf means what? They cover the food material and they form a vacuole. Clear now? That's it. And where we see that the type of food vacuoles in the case of protist. Okay, ma? In amoeba, what kind of vacuole we see? Contractile vacuole. And a tonoplast, it transport materials from which gradient to which gradient? Lower gradient to higher gradient. Against the concentration gradient or towards the concentration gradient? Against the concentration gradient. These are the points. Okay. Next, mitochondria. So they are clearly visible only when stained. What is meant by staining? Already we have studied now. Adding color to the particular cell or particular region is called as staining. So we can clearly see this mitochondria only when you stain it. Are you able to understand? Their number, shape and size per cell are variable depending on the physiological activity. Clear, Nama? See, in my body, mitochondria number will be very large. What is the reason? Because I do exercises daily. Are you able to understand? And in normal person, the amount of mitochondria will be average. And a person who is very lazy, the amount of mitochondrians or the number of mitochondrians will be very less. Are you able to understand? So mitochondria number, it is not a constant. This is one point which you should remember. Mitochondria number constant. So the person who is doing lot of physical activities in that cell of that particular person, the mitochondrial number will be very large. Are you able to understand? So it depends on the type of activity which you are doing. Okay. So therefore the number, shape and size also are variable, and it depends on the amount of the physical functions which you are doing daily. So that is why చూడండి, ఇప్పుడు జిమ్కు పోయినంత రోజులు మీరు యాక్టివ్గా ఉంటారు. ఇఫ్ యూ గో టు జిమ్ యూ కెన్ అండర్స్టాండ్. జిమ్కి పోయిన రోజులు బాగా యాక్టివ్గా ఉంటాడు మనిషి. ఎందుకంటే ద నెంబర్ ఆఫ్ మైటోకాండ్రియన్స్ విల్ బి వెరీ లార్జ్. సో చాలా ఈజీగా అది దీన్ని పవర్ హౌస్ ఆఫ్ ద సెల్ కూడా అంటాం కదా. దిస్ ఇస్ ఆల్సో కాల్డ్ పవర్ హౌస్ ఆఫ్ ద సెల్ because this is the main region where energy is produced. Are you able to understand? No? So the person who is going to gym daily, he requires more energy. No? So more mitochondria will develop, more energy will be released, and he will be active. Once you stop the gym, what happens is the mitochondria level will fall lesser than the average one. జిమ్ పోక ముందు ఒక యావరేజ్ ఉంటుంది కదా, దానికన్నా తగ్గిపోతుంది అనమాట. Because the system cannot understand what is happening suddenly. Okay, so immediately if you leave the gym after some 1 week or 2 weeks, you will feel very, very, very tired because of the reduced number of mitochondria, which is less than the average one. But again, after some days, again he will become normal. Clear? That's it. So it is sausage shaped or cylindrical. So what is the shape of mitochondrion? Sausage shaped, or you can call it as cylindrical shape, having a diameter of 0.2 to 1 micrometer. Average is 0.5 micrometer, and length varies from 1 to 4.1 micrometer. So this sizes also again depends on the activity of the person. You know? So mitochondrion is a singular form. Mitochondria is a plural. Mitochondrion is a double membrane bound structure. So how many membranes it has? It has a double membrane. Chloroplast is made up of single membrane. So questions దీనిపైన కూడా అడగచ్చు. అర్థమవుతుంది కదా. Mitochondrion is double membrane bound structure with outer membrane and an inner membrane. So it divides the lumen into two aqueous compartments. Lumen means what? The total region present inside this mitochondrion. I can call it as lumen. So it divides into two aqueous compartments, outer compartment and an inner compartment containing matrix. Are you able to understand? So there is an inner compartment in which you can find a semi-liquid which is called as matrix, ribosomes and DNA structures, all they are present in this matrix. In which compartment? In the inner compartment. Are you able to understand? So inner membrane forms a number of infoldings which is called as cristae. Okay, don't be confused between cisternae and cristae. Cisternae is the structures present in Golgi Apparatus. Cristae are the structures formed in mitochondria. Clear number? So, and they are bent towards the matrix. They are responsible for increasing the surface area. Are you able to understand? So, therefore, if you see in this figure, you can see my here. So, this membrane is called as outer membrane. Clear number? And this membrane which you see here, this is called as the inner membrane, and the gap between inner membrane and outer membrane, that is called as intermembrane space. Understood now? So here inside this inner membrane you can find this DNA also, you can find this ribosomes also, you can find this semi-liquid which is called as matrix also, and inner membrane has cristae. So these infoldings which you are seeing over here, these infoldings they are called as cristae. And what is the function of cristae? Total surface area will be increased with the help of cristae. Understood? So what are the materials which are present inside the inner membrane? Inner membrane lo emem untayi cheppandi. DNA will be present, ribosomes will be present, matrix also will be present. Okay. So the two membranes have their own specific enzymes associated with the mitochondrial function. It means inner membrane also has some enzymes, outer membrane also has some enzymes, both are which are responsible for the functioning of mitochondria. Understood, Nama? So matrix possesses a circular DNA, and where matrix is present? Matrix is present in the inner membrane. So it possesses a circular DNA which is round in shape. Few RNA molecules are also present. 70s ribosomes are present, and components for protein synthesis. So these are the different parts which are present in the matrix. వీటిపైన క్వశ్చన్ వచ్చే అవకాశం ఉంది. So matrix consists of what? Matrix means a semi-fluid type of substance. Matrix contains a circular DNA, RNA, ribosomes of which shape, which size? 70s size. Question దీనిపైన అడగచ్చు. The ribosomes which are present in the matrix, they are of which size? 30s, 50s, 70s and 60s. ఇట్లా కూడా 70s size is present. And there are some components which are responsible for protein synthesis also. Ribosome ఉంది అంటే మీనింగ్ ఏమి? ప్రోటీన్లు ఫామ్ అవుతాయి. క్లియర్ అమ్మా? Mitochondria divide by fission. This may be another important bit. Division of mitochondria is by fission process. It is not by fusion process. Okay. So mitochondria are the sites of aerobic respiration. Okay. So they produce energy in the form of ATP. So they are called as powerhouses of the cell. And what is ATP? Adenosine triphosphate is called as ATP. So this ATP is produced in mitochondria. And what kind of respiration? Aerobic respiration. It means oxygen from the blood is taken. And here the burning of food material takes place, which is also called as aerobic respiration. Clear? That's it. So we'll stop up to here. We'll continue tomorrow in the next class. Okay, last it's right. So the next concept which we are studying is plastids. Okay. So these are also organelles. They are found in all plant cells and in euglenoids also. What is meant by euglenoids? Single-celled type of bacteria. We can call it as euglenoids. No, are you able to understand? So these plastids, where they are found? They are found in all plant cells as well as they are found in euglenoids also. Okay, and these plastids where you can find in eukaryotic cells. Clear? So they are large sized, easily observable under the microscope. But mitochondria, in which condition it was observable? Mitochondria ni ela observe chestamu? Mitochondria could be observed only when stained. That was what we have studied in the last class. No? Stained means what? When we add some color to the cell, that color will be taken by the mitochondria, then only we can observe the mitochondria. But these plastids are so big that we can easily observe them under the microscope. This is the difference. And they contain some pigments. So these plastids contain some pigments also. Okay. So based on the type of pigments, plastids are of three types. So chloroplast, chromoplast, and leucoplast. So these are the three types of plastids which are present. Okay. Now these are the structures and shapes of this one. This is the chloroplast, which contains chlorophyll, and this is the chromoplast, and this is the leucoplast. Three types of plastids. So chloroplast, mainly it contains chlorophyll and carotenoid pigments. Okay, ma? So chloroplast contains what? They contains the pigment chlorophyll, and what is the purpose of chlorophyll, ma? It is responsible for photosynthesis, and some carotenoid pigments are also present. So they are helpful for trapping light energy for photosynthesis. Okay, that is the function of this plastid called chloroplast. Okay, chromoplast. Chromoplast contain fat soluble carotenoid pigments like carotene, xanthophylls, etc. They give yellow, orange, and red colors to this carotenoid, to this what you call plastids. Are you able to understand? No? So chromoplast contains what? They contain fat soluble carotenoid pigments. And what is the name of those pigments? So keratin, xanthophylls, and some another pigments are present. So what do they give? They give yellow color, orange color, and red color to this plastids. Are you able to understand? So Sima, in a chloroplast also you can find carotenoid pigments. In a chromoplast also you can find carotenoid pigments. Okay. Then what is the difference between that carotenoid pigment and this carotenoid pigment? They are fat soluble. Are you able to understand? The carotenoids which are present in chromoplast, they are fat soluble. They can melt in fat. That is the difference. Are you able to understand? And examples are keratin and xanthophyll, which is mentioned in NCERT. Clear? Leucoplast. So they are colorless plastids. They don't have any color. Okay. Of varied shapes and sizes with stored nutrients. So their colors, sizes, shapes may be different, of which of leucoplast. So plastids are colorless and they are responsible for what? They are responsible for storing the nutrients. So already I said, these plastids, what is the main function of plastids? They are responsible for photosynthesis and storing some proteins. Starting class లోనే చెప్పినాను గుర్తుందా మీకు? Plastids are responsible for photosynthesis as well as for storing some proteins. For photosynthesis, which is responsible? For photosynthesis, chloroplast plastid is responsible. For storing proteins and energy, which one is responsible? This leucoplast, they are responsible for storing proteins and energy. Are you able to understand? That's it. So they include, which include leucoplast. Leucoplasts include amyloplast, elioplast, and aleuroplast. So these are the again the three types of plastids which can be differentiated among leucoplast. Okay, ma? So amyloplast, what it will do? It will store energy in the form of starch. Example is potato. So therefore, in potato, food is stored in which plastid, ma? Food is stored in amyloplast, and amyloplast is a type of leucoplast. Leucoplast is a type of plastid. This sequence you should remember. Are you able to understand? Next, elioplast. What is the function of elioplast plastids? They store oils as well as fats. That is the function. Next, aleuroplast. What they will do? They will store the proteins. Are you able to understand? So these functions you should by heart them very carefully. So see here some of the shapes and colors of this chloroplast. These plastids are given. Chromoplast appears like this. Chloroplast. This is the leucoplast. And a leucoplast is again divided into three types. No? So this is amyloplast. This is elioplast, and this is aleuroplast. Aleuroplast is also called as protein plast also, proteinoplast also. Are you able to understand? That's it. Okay, ma? So see ma, this one. This one is also called as what? This is also called as aleuroplast. Aleuroplast and proteinoplast both are same. Fine? That's it. Next, chloroplast. Now we are studying about each of the plastids individually. Ma? So, chloroplasts are double membrane structures mainly found in mesophyll cells of the leaves. So, leaves, cells of the leaves, there are many types of cells, no ma? So, among them, where you find this chloroplast, you can find this chloroplast in mesophyll cells of the leaves. Are you able to understand? What is meant by mesophyll cells of the leaves? The smooth regions which are present in the plant, you can call them as mesophyll regions. So inside the mesophyll regions only you can find this chloroplast. Clear? So these chloroplasts, they are lens shaped, oval shaped, spherical, discoid or ribbon-like organelles. So this question also can be asked in the examination. What is the shape of the chloroplast? Chloroplast can be lens shaped also, oval shaped also, spherical shaped also, discoid also, or ribbon-like organelles also. Their length varies from 5 to 10 micrometer and width varies from 2 to 4 micrometer. Clear, Nama? And their number varies from 1 in the case of Chlamydomonas, clear, Ma? 20 to 40 per cell in the case of mesophyll cells. Are you able to understand? So, in every type of plant, you cannot see the same type of chloroplast. It depends on the type of plant which you are studying. Are you able to understand? Ma? Only one chloroplast is present in which plant? It is present in Chlamydomonas, which is also called as green algae. Are you able to understand? And between 20 to 40 chloroplasts can be found in where? In the general plants which you see, and in the leaves also, in which cells you can find them? Inside the mesophyll cells. Are you able to understand? Ma? Those cells which are responsible for photosynthesis. Fine. And this is the arrangement of this chloroplasma. You are able to see this chloroplasma inside the cells. That is how they will be arranged. So they are responsible for trapping light energy. Okay. So inner membrane of chloroplast is less permeable. This may be another bit which can be asked. Are you able to understand, ma? So which membrane of chloroplast is not highly permeable? Okay, ma? Inner membrane. Already we studied that chloroplast is a double membrane bound organelle. No? Chloroplast have how many membranes? Two membranes are present. Two layers ఉంటాయి పైన. Out of two membranes, which membrane is least permeable? Inner membrane is least permeable. So the space limited by the inner membrane of the chloroplast is called as stroma. Okay, ma? That is the total region inside this chloroplast, which is bound by the inner membrane. అంటే క్లోరోప్లాస్ట్ లోపలున్న మొత్తం ఖాళీ రీజియన్ ఏమంటాం మనము? The total region is called as stroma. Are you able to understand now? It contains a number of organized flattened membranous sacs called thylakoids. Okay, ma? Inside this stroma, that is empty region, you can find some sacs. Sacs means bag-like structures. These bags, they are called as what? They are called as thylakoids. In the figure you can see. Clear, ma? So membrane of thylakoids enclose a space called lumen. And again inside the thylakoids, that space is called as what? That space is called as lumen. Are you able to understand? So chlorophyll pigments, where they are present, they are present in the thylakoids. Inside the thylakoids, what you have? Thylakoids లోపల ఏముంటుంది? Lumen ఉంటుంది. Lumen లోపల ఏముంటుంది? In lumen only you can find this chlorophyll pigment. Are you able to understand now? So first we have, see the sequence I am writing here, ma. This sequence you should not forget. So, first of all, chloroplast. Okay, ma? Inside the chloroplast, the empty region is called what? Stroma. Inside the stroma, you can find thylakoids. Inside the thylakoids, you will have lumen. And lumen is consisting of chlorophyll pigment. Okay, ma? So this is the sequence which you should not forget. దీనిపైన కూడా వాడు ఎట్లా పడితే అట్లా క్వశ్చన్లు అడిగే అవకాశం ఉంది. Are you able to understand? So inside the figure, this empty region you are seeing, Nama, this empty region is called as stroma. Are you able to understand? No? Inside the stroma, what you have? You have this disc type of structures. No? So this disc type of structures, what you can call? You can call them as thylakoids. And this group of thylakoids, you can call them as granum. Are you able to understand now? So inside the thylakoids, what you will find? Inside the thylakoids, you will find lumen. And inside the lumen, you will find chlorophyll. Are you able to understand? That is, thylakoids is totally composed of chlorophyll pigment. And that total region, we can call it as lumen. అర్థమవుతుంది కదా sequence? So this is how these chloroplast, they are arranged. Okay? Now, we will have some more information here. So, thylakoids are arranged in stacks, which are called as grana. Okay, ma? Plural form is grana. Singular form is grana. So this group of thylakoids, what we call them? We can call them as grana. And there are many granum which are present. All of them, they are called as grana. Clear? Are you able to understand? Sima. This total one cylindrical type of structure, this is called as grana. Okay. How many granums are there? Many granums are there. So that is why they are called as grana in plural form. Okay. Or intergranal thylakoids, that also you can see. So they are flat membranous tubules called stroma lamellae connecting the thylakoids of the different granum. Are you able to understand now? Each granum and another granum, they are connected by some tubes. Are you able to understand? And what is the name of these tubes? These tubes are named as stroma lamellae. So in the figure where you can find Sima here, so these structures you are seeing now, these structures. So what is the function of these structures now? These structures, they will connect one granum to another granum. మధ్యలో ట్యూబ్స్ లాగా ఉంటాయి. Are you able to understand? It means the chlorophyll which is present in one thylakoid, it is again connected to the chlorophyll which is present in the second thylakoid, another thylakoid of the next granum. Are you able to understand? They are connected through what? They are connected to tubes which are called as stroma lamellae. This name also you should not forget. Okay, ma? So stroma contains small double-stranded circular DNA molecules. So along with this granum, the empty space called stroma also consists of DNA molecules also, ribosomes also, enzymes also for what? For the synthesis of carbohydrates as well as proteins. అర్థమవుతుంది కదా? Stroma is the space where it is present. Which region is called stroma? Stroma అనేది ఏమని అంటాం? The region which is present inside the chloroplast is called as stroma. And inside the stroma, you can find grana also, which is a stack of thylakoids, which is a pile of thylakoids. And some more empty space is there, no? In that empty space, again, you can find this double-stranded DNA molecules also. Here also, you can find ribosomes. And some other enzymes are also present which are responsible for the production of carbohydrates as well as proteins. Okay, ma? See, ma, where is the arrangement of ribosomes? Ribosomes are present in many regions. It is present in cytoplasm also. It is present on rough endoplasmic reticulum also. Clear, ma? They are present inside this stroma of this chloroplast also, and they are present on the surface of the nucleus also. In eukaryotic cell, we will study nucleus, no? On the nucleus also, you will find ribosomes. And what is the function of ribosomes? Ribosomes function aiming production of proteins, synthesis of proteins. Already we have studied, no? That's it. Okay, ma? So the ribosomes of the chloroplast are smaller than the cytoplasmic ribosomes. So this may be another important bit which generally you will not concentrate. Clear, ma? So the ribosomes which are present inside this stroma and the ribosomes which are present on the endoplasmic reticulum, they are not one and the same. Their sizes are different. Okay, ma? So 70s is the size which is present where? Which is present in what you call this grana. Sorry, beside the grana or inside the stroma. And 80s is the size of the ribosomes present in endoplasmic reticulum, or you can call it as cytoplasm. Are you able to understand, ma? That's it. So sizes, they are not same. Sizes are different. Okay, ma? Next. So, okay. So these are next ribosomes we are studying. Ma. These are non-membranous granular structures composed of ribonucleic acid and proteins. So ribosomes basically they contains what, ma? They contains RNA and they contain proteins. Are you able to understand? And the most important point is they are non-membranous granular structures. So ribosomes does not have any membrane. Are you able to understand, ma? No. So they consist of basically ribonucleic acid RNA and they contain proteins also. Are you able to understand, ma? So George Palade first observed ribosome in 1953. Are you able to understand? And these small dot-like structures which you are seeing, these you can call them as ribosomes. Some of the ribosomes are present on rough endoplasmic reticulum. Some of them they are present in cytoplasm. Some of them they are present in inside the chloroplast, and some of them you can find on nucleus also. Clear, Nama? So eukaryotic ribosome has two subunits. So the ribosomes which are present in eukaryotic cells, the cells which have nucleus. So it is basically made up of two subunits. Okay, ma? One is 60 years and the other one is 40 years. Okay, they together form 80 years. It means 60 years sized ribosome and 40 years sized ribosome. Both of them they give rise to 80 years sized ribosome. Are you able to understand, ma? That is, so this is the 40 years and this one is the 60 years. Both of them they combine together to form the 80 years ribosome. Understood, no? That's it. And 80s ribosomes where they are present, 80s ribosomes they are present on rough endoplasmic reticulum or inside the cytoplasm. Are you able to understand, ma? 70s ribosomes where they are present inside the chloroplast, inside the stroma. Clear? That's it. Next, cytoskeleton. Are you able to understand, ma? So a network of filamentous proteinaceous structures present in the cytoplasm is called as cytoskeleton. Are you able to understand, ma? So they are like some irregularly arranged tubes, ma. Are you able to understand? We know what is meant by skeleton, no? Skeleton means the structure which gives shape and size to the body is called as skeleton. So here also some filamentous tubes are present, and all of them filamentous tubes we can call them as cytoskeleton. And they are made up of what? They are made up of proteins. That is why it is called as proteinaceous. Okay. So it provides mechanical support, motility, maintenance of the shape of the cell. So what is the function of any skeleton, ma? Maintaining shape and size only, no? Human body is able to maintain its shape and size only because the skeleton which we have. Skeleton లేకపోతే మనం గుండ్రంగా సాలిడ్ స్పియర్ లాగా తయారవుతాం అనమాట. Are you able to understand? So here also inside the cell also cell is able to maintain its shape and size because of these filamentous proteins, which is called as cytoskeleton. So this total structure which you are seeing, Numa, this total yellow color structures which you are seeing, this total yellow color structures they are called as cytoskeleton. Are you able to understand? Okay. Okay. So next we study about cilia and flagella, which are responsible for motion of a cell. So they are hair-like outgrowths of the cell membrane. Okay, ma? So where you can find this cilia and flagella, ma? Cilia and flagella you can find on the outer surface of this cell, on the outer membrane of the cell. Clear, ma? So cilia, what is the function of cilia? Small structures which work like oars, okay, ma? Causing the movement of cell or surrounding fluid. That is called as cilia. Are you able to understand? Oars means what? For rowing a boat, you need oars, no? Huh? So this cilia also they function like oars. So they are responsible for movement of the cell, or simultaneously you can say they are responsible for movement of the liquid in which the cell is present. If a cell moves, you can say motion of the cell is present. If the surrounding fluid flows, then also you can say cell is moving. Okay, that's it. Flagella. They are longer, responsible for cell movement. Flagella of prokaryotes and eukaryotes are structurally different. So the structure of flagella present in prokaryotes and the structure of flagella present in eukaryotes, they are not one and the same. They are different. Are you able to understand, ma? So basically, cilia and flagella, what they will do? Both of them, they are responsible for movement of the cell. Are you able to understand, ma? That's it. Okay. So cilium and flagellum, they are singular forms, are covered with plasma membrane. Okay, ma? So question can be asked on this presence of membrane and absence of membrane also, ma. Okay. Chloroplast, double membrane structure. Ribosomes does not have any membrane. Cilium and flagellum, they have single membrane structures. Okay, ma? సో ఈ కింది వాటిలో దేనికి మెంబ్రెయిన్ ఉండదు, ఇలా అయినా క్వశ్చన్ని ఫ్రేమ్ చేయొచ్చు. ఓకే, సో ప్రతి డీటెయిల్ని మీరు ముందు వాటితో కంపేర్ చేసుకుంటూ చదవాలి. బయాలజీ చదివే పద్ధతి అదే. ఓకే, సో cilium and flagellum are covered by plasma membrane. How many membranes are there? Single. Only single plasma membrane is present. Okay, ma? So their core has many microtubules running parallel to the long axis. Okay, core. And what is the name of the core? Axonym. Axonym has many microtubules. Microtubules means what, ma? Small tubes are present. Inside cilium and flagellum, you can find again small tubes which are running throughout the cilium and flagellum. For example, I can draw the figure like this. Clear, Nama? So this is a flagellum you consider. So inside this flagellum, again you can find small this type of tubes which are running. Are you able to understand, ma? And what they are called? They are called as microtubules. So inside the cilium and flagellum, again you can find small tubes which are called as microtubules. Okay, ma? And what is the name of the core? Core name, core pair empire. Core means central region. Central region. Axonym is the name of the central region. So axonym has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of central tubules. This is called 9 plus 2 array. Are you able to understand, ma? So in the figure here you can see, ma. In the figure, so these are the two central microtubules. These two are into central microtubules. Are you able to understand, ma? So this is first one and this is the second one. And surrounding them we have 9 pairs of tubules. Okay, 9 pairs means totally how many? 18 tubules are present. Are you able to understand? So in the figure you can draw them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 like this. Totally 18 are present. Are you able to understand, ma? And in the middle we have only 2 tubules which is present. So that is why this system, what you can call them as 9 plus 2 array. So where you are seeing this arrangement, you are seeing this arrangement inside the cilia and flagella. ఓకే, అవే చాలా చిన్న బేసికల్గా కంటికి కనిపించవు, దాంట్లో మళ్ళీ ఇంత అరేంజ్మెంట్ ఉందంట. క్లియర్ నమ్మా? సో దిస్ ఈస్ ద అరేంజ్మెంట్ ఆఫ్ దిస్ tubules. So this arrangement we can call them as 9 plus 2 array. Now see here some organelles are given. Let us see. Okay. So see. So this one is called as radial spoke. Okay. This tube which you are seeing here, this is called as radial spoke. Kanipistundi kada? Okay. And these tubules, they are called as central microtubules. So how many central microtubules are present? Two central microtubules are present. Okay. And here you have interdoublet bridge. Interdoublet bridge is present. So what is the function of this interdoublet bridge? This green color structure which you are seeing here, that is called as interdoublet bridge. That is responsible for connecting two tubules, outer tubules. Are you able to understand? For connecting one pair of tubule and another pair of tubule. So this interdoublet bridge is responsible. Clear, Nama? Then we have peripheral microtubule. Peripheral microtubules means what? The 9 pairs of tubules which are present, they are called as peripheral microtubules. And there is a plasma membrane and there is a central sheath. Okay. So these central microtubules, they are surrounded by a layer. And what is the name of that layer? That layer is called as central sheath. అక్కడ రెడ్ కలర్ షేప్స్ కనిపిస్తా ఉన్నాయి కదా మీకు. సో దోస్ రెడ్ కలర్ స్ట్రక్చర్స్ దే ఆర్ కాల్డ్ as central sheath. Are you able to understand, ma? So we will study in detail about how connections are present here. Here we will study. So the central tubules are connected by bridges and are enclosed by a central sheath. Okay, ma? So central tubules are connected by bridges means what? Which bridge is present here? Okay, ma? So central tubules, Sima, they are both of them, they are connected by this bridge also, and peripheral tubules are there, no, surrounding tubules. They are also connected to the central one by these bridges. Are you able to understand what is happening? Central microtubules are also connected, and peripheral microtubules, they are also connected to central ones, again with the help of some bridges. Are you able to understand? That's it. Okay, ma? Central tubules are closed by central bridges and are enclosed by central sheath. So this red color region, that you can call it as central sheath. So it is connected to one of the tubules of each peripheral doublet by a radial spoke. Are you able to understand, ma? So there are 9 radial spokes. It means central sheath, that red color region is there too. That central sheath will be connected to each peripheral tubule again by a spoke, again by a tubule. How many peripheral tubules are there? 9 peripheral tubules are there. So therefore, how many spokes should be present? 9 spokes should be present. So these 9 spokes, they will be connected to the central sheath. And where is this central sheath? Central sheath is surrounding this central microtubules. Are you able to understand? This much of arrangement is present. The peripheral doublets are also interconnected by linkers. So the doublets, how many doublets are there? 9 doublets are there. These 9 doublets are again connected by some spokes, which you can call them as linkers. And where is that linker? So here you are seeing, no, these green color structures you are seeing, no, they are the linkers. Are you able to understand? They are connected by the linkers. Okay. So cilium and flagellum emerge from centriole-like structure called basal bodies. Are you able to understand, ma? So cilium and flagellum, from where they will start? They will start from the basal body, centriole-like structure, which is called as basal bodies. So from the basal, already we studied, no, ma? This flagellum, it can be divided into 3 parts. Okay, basal body, hook, and a filament. So similarly here also we have a basal body. So from the basal body only this cilium and flagellum, they will arise. Okay. So next, centrosome. Now next we are studying about centrosomes and centrioles. Clear, Nama? So okay, Sima, group of centrioles, we can call them as centrosome. First, this point we should remember. Okay. So group of centrioles is called what? It is called as centrosome. Okay. And how they are arranged, ma? See here, arrangement is given in the figure. They are arranged like this. It means one centriole is like this, another centriole will be like this. Okay. So this total perpendicular centrioles, what we call them? We call them as centrosomes. Are you able to understand? So this is one tubule which is present, this is another tubule which is present, which are perpendicular to each other, and that combination, what we call, that combination is called as centrosome. Are you able to understand? Now let us see. So centrosome is an organelle containing two non-membrane bound cylindrical structures called centrioles. Are you able to understand, ma? So centrosome is made up of non-membrane bound structures. It means centrioles does not have any membrane. So this is another important point. Are you able to understand, ma? Centrioles paina membrane undadu. Okay. So they are surrounded by pericentriolar materials. Okay, ma, what is the name of the materials which these centrioles they are surrounded by? Those materials they are called as pericentriolar materials. So centrioles lie perpendicular to each other. Already we discussed, no? So here the angle between two centrioles is how much? It is 90 degrees. Centrioles lie perpendicular to each other. They are made up of 9 evenly spaced peripheral fibrils, which are made up of a material which is called as tubulin. Are you able to understand, ma? It means how I can say that. So this is uh, one fibril, second fibril, third fibril, fourth fibril, fifth fibril, sixth one, seventh one, eighth one, and finally ninth one. So like this, how many peripheral fibrils will be present? 9 peripheral fibrils will be present. And each fibril, again, it is made up of a triplet. Triplet means what? 3 tubules, they make one fibril. 9 fibrils, they will make one, what you called centriole. And 2 centrioles, which are perpendicular, they will make a centrosome. So this is the arrangement. Are you able to understand, ma? See, ma, in the figure you can see here. So here you have a triplet, no? ఇక్కడ ట్రిప్లెట్ కనిపిస్తుందా మీకు? ట్రిప్లెట్ మీన్స్ వాట్? 3 fibrils, 3 tubules which are connected together, they are called one triplet. Okay, so that triplet we can call it as what? We can call it as a fibril. Are you able to understand, ma? And like that, how many fibrils are present? 9 fibrils are present. Then a cylindrical shape will be formed, no, ma? That cylindrical shaped tube we can call it as centriole, and 2 perpendicular centrioles is called as centrosome. That is the arrangement. Clear? So next year, each of the peripheral fibril is a triplet. Triplet means what? 3 tubes or 3 tubes we can say. 3 tubes is called as triplet. So the adjacent triplets also they are linked with each other. అర్థమవుతుందా ఫిగర్ లో అరేంజ్మెంట్ ఎలా అవుతున్నాయో అవి? 3 triplets are present. Sorry, each fibril is made up of a triplet, and 9 fibrils are present. 9 fibrils will make one centriole, and perpendicular centrioles will make a centrosome. That is the arrangement. Clear, Nama? That's it. So the central part of the centriole is also proteinaceous and is called the hub. It means inside the centriole, that the tube-like structure, it is completely made up of proteins. అర్థమవుతుంది కదా? And that proteinaceous region, what we call, that is called as the hub, which is connected with tubules of the peripheral triplets by radial spokes, which are made up of protein. Are you able to understand, ma? And inside this hub also, you can find spokes which are emerging from the fibrils. Are you able to understand? It means, for example, this will be the shape, Noma, approximately. From each triplet also, again, you can find some spokes which are coming out. Okay, and all these spokes, they are connected inside the hub. And what is the hub? The region inside the centrosome, which is made up of proteins. Clear? So the centrioles from the basal body of the centrioles form the basal body of cilia and flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cell. So what is the function of a centriole, ma? What is the function of centriole? They form the basal body. And what is the function of basal body? From this basal bodies only cilia and flagella they will develop. Are you able to understand, ma? And one more function is they are responsible for forming the spindle fibers also. And where these spindle fibers they are responsible? they are having function in the case of cell division. Okay, ma? When cells divide, we require fibers which is called as spindle fibers. And for the formation of spindle fibers, centrioles are responsible. And for the formation of basal bodies also, centrioles are responsible. Are you able to understand, ma? So two functions of centrioles. One is formation of basal body. Second one is forming of the spindle apparatus. Clear? That's it. Nucleus. So nucleus was first described by Robert Brown. The materials of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Okay, ma? So inside the nucleus, if you stain the nucleus ki koncham color add chesnaru anukondi. Okay, ma? You will find some thread-like structures which are present inside the nucleus. Those thread-like structures, we can call them as chromatin. Are you able to understand? Ekkada unnaya thread-like structures? So here you are seeing this thread-like structures, no ma? Okay. So these thread-like structures, they are called as chromatin. So when you can see them only when you dye the nucleus. When you add some color to the nucleus, then you can find that thread-like structures. They are called as chromatin. And these chromatin only, they are responsible for the formation of chromosomes. Chromosomes ఎక్కడి నుంచి ఫామ్ అవుతాయి? Chromatin నుంచి ఫామ్ అవుతాయి. Clear nama? So normally a cell has only one nucleus. Okay, ma? Some cells have more than one nucleus also. This is one important bit. So most of the cells, they have only single nucleus only, ma? But in some cases you can find more than one nucleus also. So some mature cells, they lack nucleus. It means cells after they get matured, some cells, so their nucleus may be absent also. Clear nama? So example, mammalian RBC, that is the RBC which is present in mammals, when they mature, nucleus will be absent. Okay. And sieve tube cells of vascular plants. We know what is meant by sieve tube cells, Nama? In the plant cells we have studied. Okay. Vascular plants means what? Vascular bundles means what? Xylem and phloem only, no? So surrounding the xylem and phloem, you can find this sieve tube cells. So therefore, in two types of cells, what you call nucleus will be absent. What are the two types of cells? One is mammalian RBC when it matures, and the second one is sieve tube cells. When these cells also mature, you don't find the nucleus. Clear, ma? So these are two important ones which you should remember. Okay. So the interface nucleus contains, that is the structure of nucleus contains nuclear envelope. Nuclear envelope means what? The cover on the nucleus is called as nuclear envelope. It is made up of two membranes. Clear? Nuclear envelope has how many membranes? Two membranes are present. So then a liquid type of material is present which is called as nuclear matrix. It is also called nucleoplasm. Okay, ma? And chromatin fibers are present, and inside the nucleus again you find a structure which is called as nucleolus. So these are the different regions which are present inside the nucleus. Clear, nama? So in the figure you can see here. So nuclear pores, it means small holes are present on the nucleus. On the nucleus you will find small holes. So from these holes only these chromosomes they come out. Are you able to understand? RNA molecules also they come out of these holes. Chinna chinna holes ఉంటాయి nucleus పైన. Clear nama? And ribosomes are also present on the layers. Endoplasmic reticulum. Clear nama? Then inside the nucleus you will find another structure which is called as nucleolus. And this dyed thread-like structures they are called as chromatin. And outer structure of this nucleus is called as nuclear envelope. So these are the different parts. Okay. Now let us study each of them in detail. So nuclear envelope. So double layered membrane. This is a very important point. Clear, ma? So the envelope, the cover which is present on the nucleus, it is made up of two membranes. That is why it is called as double membranous, with a space between 10 to 15 nanometer called the perinuclear space. Okay, ma? So what is the meaning of this one? Inside the membranes also you will find a space. First membrane, second membrane ఉంటాయి కదా, వాటి మధ్యలో కూడా ఒక స్పేస్ ఉంటుంది. And what will be the length of the space? It will be 10 to 50 nanometer. Are you able to understand, ma? And that space, what you call that space is called as perinuclear space. Are you able to understand? See in the figure you can see here. So this outer structure, this is called as outer membrane, and you will find an inner membrane also. Okay, and the region which is present between outer membrane and inner membrane, that is called as perinuclear space. So it is a barrier between materials in the nucleus and in the cytoplasm. It means these layers, they are protecting the intermixing of the materials present in cytoplasm and materials which are present in nucleus. అవి రెండు మిక్స్ అవ్వకుండా ఈ మెంబ్రేన్ కాపాడుతూ ఉంటుంది. ఓకేమా. Outer membrane usually continues with the endoplasmic reticulum and also bears ribosomes with it. Already we have studied, Nama. Surrounding the nucleus, what you will find? You will find endoplasmic reticulum. Which endoplasmic reticulum? Rough endoplasmic reticulum. On the rough endoplasmic reticulum, you can find ribosomes. And surrounding the rough endoplasmic reticulum, you will have smooth endoplasmic reticulum. There is no ribosomes on smooth endoplasmic reticulum. Clear, Nama? So nuclear envelope. Nuclear envelope means what? The cover of this double membrane nucleus. Okay, ma? Has minute pores. Pores means what, ma? Holes. Holes are present. Formed by the fusion of both membranes. It means these holes, they pass through first membrane also, they pass through second membrane also. ఈ హోల్స్ ఉంటాయి కదా పైన ఎన్వలప్ పైన, అవి ఫస్ట్ మెంబ్రెయిన్ ని సెకండ్ మెంబ్రెయిన్ రెండిట్లోకి చొచ్చుకొని పోయి ఉంటాయి ఆ హోల్స్. ఆర్ ఏబుల్ టు అండర్స్టాండ్? So these are the passages for the movement of what? For the movement of RNA. So RNA molecules, they come inside and go outside of this nucleus because of these holes, pores which are present on the envelope. And proteins between nucleus and the cytoplasm. So it means RNA molecules are also exchanged. Proteins also they are exchanged. Okay, ma? Next, chromatin. What is meant by chromatin, ma? So the structures, the thread-like structures which you see when you dye this cell is called as chromatin. So these are a network of nucleoprotein fibers. It means they are made up of proteins only. ఇవి కూడా proteins తోనే తయారు చేయబడి ఉంటాయి. So it contains DNA and basic proteins like histones, non-histone proteins as well as RNA. Okay ma? So chromatins లో ఏమేమి ఉంటాయి? DNA will be present, RNA will be present, histones will be present, non-histone proteins are also present. So during cell division, chromatins condense to form chromosomes. So these chromatins itself, they combine to become what, ma? They combine to become chromosomes. Are you able to understand, ma? That's it. So chromosomes ఎక్కడి నుంచో ఫామ్ కావు. ఎక్కడి నుంచి ఫామ్ అవుతాయి? ఈ క్రోమాటిన్స్ నుంచే ఫామ్ అవుతాయి. క్లియర్ మా? నెక్స్ట్ న్యూక్లియోలస్. వాట్ ఈస్ మెant by nucleolus? Inside the nucleus again you will find a spherical structure. This spherical structure is called as nucleolus. Are you able to understand, ma? So one or more non-membranous spherical bodies. Are you able to understand, ma? So nucleolus may be single nucleolus also may be present. More than one nucleus also may be present inside the nucleus. Nucleus కూడా ఒకటి ఉండొచ్చు, ఒకటి కన్నా ఎక్కువ ఉండొచ్చు. అర్థమవుతుంది కదా? Inside the eukaryotic cell, you may find a single nucleus also, more than one nucleus also. And inside the nucleus again you can find single nucleolus or more than one nucleolus. Are you able to understand, ma? So again it is it continues with the nucleoplasm. What is meant by nucleoplasm? The semi-fluid type of liquid present inside the nucleus is called as nucleoplasm. And inside this nucleoplasm you can find this nucleolus. అర్థమవుతుంది కదా? So it is a site for what? It is a site for ribosomal RNA synthesis. Okay, ma? So RNA production, where you can find RNA production, you can find in nucleolus, which is present inside the nucleus. Fine. And which kind of RNA synthesis, ma? Ribosomal RNA synthesis. It means the RNA which is present in ribosomes, that is manufactured inside the nucleus. Fine. That's it. Okay, ma? Chromosomes. So human cell has about 2 meters long DNA distributed among its 23 pairs of chromosomes. So human cell, each cell has how many chromosomes, ma? 23 pairs are present. It means totally 46 chromosomes are present. Are you able to understand, ma? And this DNA, what is the length of this one? It is approximately 2 meters. సెల్లే మనకు సరిగా కనిపించదు కంటికి, దాంట్లో మళ్ళీ 2 మీటర్ల లెంత్ ఉంటే డిఎన్ఏ సర్కులర్గా డిస్ట్రిబ్యూట్ అయ్యి ఉంటుంది. Are able to understand ma? It means a very very huge amount of information is present in each cell of the body. ప్రతి సెల్లులో కూడాను విపరీతమైన ఇన్ఫర్మేషన్ ఉంటుంది. నువ్వెవరు, నీ పేరేం పేరు, మీ నాయన పేరు ఏం పేరు, మీ నాయనకి ఎట్టాటి అలవాట్లు ఉన్నాయి, మంచి అలవాట్లు ఉన్నాయి, దరిద్రమైన అలవాట్లు ఉన్నాయి, అవన్నీ కూడా ఈ డిఎన్ఏలో స్టోర్ అయి ఉంటాయి. అది ఎక్కడ ఉంటుంది? ప్రతి సెల్లులో కూడా ఉంటుంది అనమాట. అరేబుల్ టు అండర్స్టాండ్ అంటే ఒక సెల్లును మీరు కరెక్ట్గా పట్టుకొని దాని ఇన్ఫర్మేషన్ తీసినారంటే బయటికి మీ చరిత్ర అంతా చెప్తుంది అది. So that much amount of information is stored in each cell of human body. Are able to understand? So every chromosome has a primary construction, which is called as a centromere, on the sides of which disc-shaped structures called kinetochores are present. Are able to understand ma? So each chromosome, every chromosome has a constriction. Constriction ante me oka ఏమని చెప్తాం మనము? Consider this is a pipe. ఓకేమా? ఈ పైప్ మధ్యలో మీరు ఒక ఏదన్నా గట్టిగా కట్టేసినారు అనుకోండి, అప్పుడు ఆ షేప్ ఇలా తయారవుతుంది కదా. దట్ టైప్ ఆఫ్ స్ట్రక్చర్ ఈస్ కాల్డ్ యాజ్ వాట్ యూ కెన్ కాల్ ఇట్ యాజ్ ఎ కన్స్ట్రక్షన్. ఓకేమా? మధ్యలో తాడుతో గట్టిగా బిగించేస్తే ఆ రీజియన్ని మనం కన్స్ట్రక్షన్ అంటాం. Are able to understand? So each chromosome has a constriction. So this is the constriction. Okay ma? So this total structure is called a chromosome, and in the middle you have some bound structure that is called as a constriction. Okay, and on the constriction what you will have, you will find a disc shaped structure called as kinetochores. Okay, so here you are seeing this small green color structures you are seeing noma. So this small green color structures, they are called as kinetochores. Ikkada yellow color structures kanipista unnayi chudandi. These yellow color structures, they are called as kinetochores. Are you able to understand? That's it. Okay ma? So next, based on the position of this what you called constriction, okay ma, which is also called a centromere, we are dividing this chromosomes into 4 types ma. So ee madhyalo constriction untundi kada, which is called a centromere. Where this centromere is present, depending on that again chromosomes we can divide into 4 types. Are you able to understand ma? Centromere may be present exactly in the middle, centromere may be slightly away from the middle, centromere may be at a very large distance from a middle, centromere may be exactly at what one end of the chromosomes. So depending on that we are dividing chromosomes into 4 types. Clear ma? Metacentric chromosome. So middle centromere forming 2 equal arms of the chromosome. Are you able to understand? It means, see, I will draw the shape here. So what is this region? This is called as centromere. No? The constriction which is present is called as centromere. Okay. So if that centromere is dividing the chromosome into 2 equal lengths, it means the length of this one and your length of this one, if they are equal, then it is called as metacentric chromosome. Submetacentric chromosome. Submetacentric means it is not exactly in the middle. One arm is slightly bigger, one arm is slightly smaller. Then it is called as submetacentric. Acrocentric chromosome means what? One arm is very small, one arm is very big. It means, how can you say the arrangement of this one? So these are the 2 arms ma, and the next 2 arms are like this. Are you able to understand? If 2 arms are very small no, and these 2 arms they are very big, then they are called as acrocentric chromosome. And telocentric chromosome. Telocentric chromosome means what? So this centromere, it is present at one end of the chromosome. It means there is no division of small arm and big arm. Are you able to understand? Then this type of arrangement you can call it as telocentric chromosome. And here there is one more point. Some chromosomes have non-staining secondary constrictions also at a constant location. It is called a satellite. So some of the chromosomes, they have another constriction. Rendu constrictions untai. Okay ma? And that second constriction, what you call? Second constriction you can call it as a satellite. Are you able to understand? So this is the second constriction which you can find, and this is called as the satellite. Are you able to understand? That's it. Okay ma? Then microbodies. Microbodies, these are just membrane bound minute vesicles which contain various enzymes. Okay, ma? They are present in plant cells also, they are present in animal cells also. And the point which you should remember is what? It is a membrane bound organelle. క్లియర్ నమ్మా. సో ఏవి మెంబ్రెయిన్స్ ఉన్నాయి, ఏవి మెంబ్రెయిన్స్ ఉండవు, అవన్నీ మీరే లిస్ట్ చేసుకోవాలి ఫస్ట్. రేబుల్ టు అండర్స్టాండ్. మీరు ఎక్కడైతే ఆలోచించారో అక్కడ నుంచి క్వశ్చన్ ఇస్తాడు వాడు. క్లియర్. సో దీస్ ఆర్ మెంబ్రెయిన్ బౌండ్ minute vesicles which contain various enzymes. Enzymes are present inside these microbodies. And where they are present, you can find them in plant bodies also, you can find them in animal bodies also. In prokaryotes, we have studied inclusion bodies. No? Prokaryotes lo etuvanti bodies nemantamu, inclusion bodies antam. In eukaryotes, we can call them as microbodies. That is the difference. Okay. So then, this is the end of this chapter. No? We have small differences which we need to see once again. So plant cell and animal cell. Cell walls, they are present in plant cell, they are absent in animal cell. Plastids are present in animal cell, plastids are absent. Large central vacuole is present in plant cell. And what is the function of vacuole? Already we have studied, Noma. So vacuole is a region where 90% of the plant cell is covered. Okay, so all the waste material, everything, it will be present inside this vacuole. Okay, and materials, they were transferred against the concentration gradient. anta chadunam kada manam. Whereas in animal cell, many small vacuoles are present. In the plant cell, there is only single vacuole which covers up to 90% of the volume of the cell. In animal cell also we have vacuoles, but not single vacuole. Small vacuoles are there, but many vacuoles are present in animals. That is the difference. Okay, ma? Centrioles are absent, whereas centrioles, they are present inside the animal cell. So these differences are very important. Clear? You should by heart them. Next, already we have studied. Okay, ma? Next, difference between prokaryotic cells and eukaryotic cells. So generally smaller, eukaryotic cells are generally larger. Genetic material in the form of nucleoid, because in prokaryotic cell, nucleus is not present, no ma? There is a central region which is called as nucleoid, and total information of prokaryotic cell is present in the nucleoid itself. So, but it is present in the form of nucleus, genetic material in the form of nucleus. Nuclear membrane is absent. Nucleus lenappudu membrane ekkadi nunchi vastundi? Membrane is present. Membrane bound organelles, they are absent, and membrane bound organelles, they are present. So circular DNA. DNA is circular in shape, and here we have a linear DNA. Clear, no? Ribosomes are 70 years. So, but here they are 80 years. But inside the what you call plastids. Plastids means which plastids? Chloroplast. Okay, ma? Inside the chloroplast, what is the size of ribosomes, ma? 70 years ribosomes. Also in mitochondria also you can find 70 years ribosomes. So 70 years ribosomes you can find in animal cells. And in animal cells, where you find? Inside the chloroplast as well as mitochondria. Remaining all the ribosomes, they are 80 years ribosomes. Clear? That's it. That completes our chapter called Cell, the Unit of Life. Okay? We will continue in the next class.